Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The history of oncology is largely a chronicle of assaults upon the malignant cell. For over a century, the primary therapeutic modality, radiation, and chemotherapy shared a common tactical objective: the direct eradication of tumor tissue. This strategic orthodoxy, while yielding considerable success, viewed the patient's own biological systems as a passive battlefield, a landscape to be cleared of cancerous invaders. The immune system, in this context, was often seen as a collateral casualty or, at best, a mystifyingly inept bystander. The idea of enlisting it as an ally, a concept known as immunotherapy, flickered at the periphery of mainstream medicine for decades, a field rich in promise yet poor in reproducible, durable outcomes. Earlier efforts, such as William Coley's use of bacterial toxins in the late 19th century to provoke a general immune fever, were tantalizing but largely uncontrollable, their mechanisms opaque. The prevailing assumption was that the immune system simply failed to recognize cancer, or that its response was intrinsically feeble. The intellectual revolution ignited by the work of James Allison and Tasuku Honjo, culminating in the therapeutic paradigm of immune checkpoint therapy that gained definitive clinical validation around 2010, proposed a radically different and far more profound diagnosis. The immune system was a fully equipped and capable fighting force. The problem was that cancer had discovered its off-switches. The genesis of this paradigm shift lies in the fertile ground of basic immunology research of the 1980s and early 1990s. The discovery of the T-cell receptor and the elucidation of the two-signal model for T lymphocyte activation provided the essential vocabulary for understanding immune responses. This model posited that full activation of a T-cell required two signals. The first came from the T-cell receptor recognizing a specific antigen presented by another cell, and the second came from a costimulatory molecule. The most well-characterized of these costimulatory pathways involved the cluster of differentiation 28, or CD28, receptor on the T-cell binding to its ligands, B71 and B72, on the antigen-presenting cell. This was the go signal, the accelerator pedal for the immune response. Within this framework of activation, a molecule discovered in 1987, named cytotoxic D-lymphocyte-associated protein 4, or CTLA4, presented a puzzle. It bore a striking structural resemblance to the stimulatory CD28 receptor and also bound to the same B7 ligands. The logical, prevailing hypothesis was that CTLA4 must therefore be another costimulatory molecule, another accelerator. It was here that James Allison, working at the University of California, Berkeley, made his seminal intellectual leap. Allison's laboratory, through a series of elegant experiments, developed the counterintuitive hypothesis that CTLA4 functioned in the exact opposite manner. It was an inhibitory receptor, a molecular break. His team demonstrated that CTLA4 possesses a much higher affinity for the B7 ligands than CD28 does, allowing it to effectively outcompete the accelerator and shut down the T-cell response. This was a crucial mechanism for maintaining self-tolerance and preventing autoimmunity, a way for the immune system to police itself. Allison's profound insight was to wonder what would happen if one deliberately cut this break line in the context of cancer. Simultaneously, and working along a parallel intellectual trajectory in Japan, the immunologist Tasuku Honjo at Kyoto University was investigating a different molecule. In 1992, his laboratory identified a gene that was induced during the process of programmed cell death, or apoptosis. They named the protein it encoded programmed cell death protein 1, or PD1. Its initial characterization suggested a role in terminating immune responses, yet its precise function remained elusive for nearly a decade. Honjo's group meticulously unraveled its biology, demonstrating through knockout mouse experiments that animals lacking PD1 were prone to developing spontaneous autoimmune diseases, providing compelling evidence of its inhibitory function. Further research from his lab, along with crucial contributions from the laboratories of Gordon Freeman and Arlene Sharp at Harvard, identified the ligands for PD1 programmed death ligand 1 and programmed death ligand 2, or PDL1 and PDL2. A critical discovery was that PDL1 is expressed broadly on many cell types throughout the body and can be upregulated on the surface of cancer cells themselves. This revealed a distinct and insidious mechanism of immune evasion. While the CTLA4 break functions early in the immune response, primarily in lymph nodes where T cells are first activated, the PD1 break operates later, within the tumor microenvironment itself. A tumor cell, by displaying PDL1 on its surface, could directly engage the PD1 receptor on an infiltrating T cell and command it to stand down, creating a zone of immune privilege. Honjo had uncovered an independent though equally powerful brake on the anti-tumor immune response. The translation of these fundamental biological discoveries into a therapeutic modality represents one of the most dramatic bench-to-bedside stories in modern medicine. Allison relentlessly championed his concept, proposing that an antibody designed to block CTLA-4 could unleash a potent and pre-existing anti-tumor T-cell response. The idea faced considerable skepticism from a pharmaceutical industry accustomed to drugs that directly kill cells. The concept of a therapeutic that functions by inhibiting an inhibitor was a difficult proposition. Eventually, the biotechnology company MedX developed a humanized anti-CTLA-4 antibody, which would become known as ipilimumab. The clinical validation of Allison's hypothesis arrived with breathtaking force in 2010, with the publication of a landmark Phase 3 clinical trial in patients with metastatic melanoma. For this disease, which had seen almost no therapeutic progress in decades, ipilimumab demonstrated a clear overall survival benefit. While the response rates were modest, the responses themselves were unprecedented in their durability. A fraction of patients, previously considered terminal, experienced long-term remissions lasting for years, their survival curves forming a distinctive plateau that hinted at a functional cure. This trial was the definitive proof of concept. It showed that releasing a single immunological brake could, in some patients, completely alter the course of a fatal malignancy. The work on the PD-1 pathway followed a similar trajectory. Antibodies developed to block either PD-1, such as nivolumab and pembrolizumab, or its ligand PD-L1, entered clinical trials in the early 2010s. The results were even more startling. These agents produced higher response rates than anti-CTLA-4 therapy and, critically, across a much wider range of cancer types, including non-small cell lung cancer, renal cell carcinoma, and Hodgkin's lymphoma. Furthermore, their side effect profile, while still significant, was generally more manageable. The year 2010 sits as a watershed moment when the clinical data became undeniable, transforming checkpoint inhibition from a fascinating biological theory into the fourth pillar of cancer treatment. The legacy and evolution of checkpoint therapy continue to unfold with remarkable speed. The initial success with single-agent therapies logically led to the exploration of combination strategies. The combination of anti-CTLA-4 and anti-PD-1 therapies, for example, has been shown to produce synergistic effects and deeper responses in certain cancers, such as melanoma, albeit at the cost of increased toxicity. This reflects the distinct, non-redundant roles of the two pathways. Blocking the CTLA-4 brake in the lymph node primes a broader T-cell response, while blocking the PD-1 brake in the tumor allows those activated T-cells to execute their function more effectively. The success of these first-generation checkpoint inhibitors has catalyzed an explosion of research into the tumor microenvironment and the myriad other inhibitory and stimulatory pathways that govern immune function. Researchers are now actively investigating additional checkpoint molecules, such as lymphocyte activation gene 3, or LAG3, and T-cell immunoglobulin and mucin domain-containing 3, or TIM3, as new therapeutic targets. The central challenge guiding the field today is understanding the mechanisms of resistance. A substantial portion of patients derives no benefit from these therapies, and many who initially respond eventually relapse. This has driven a global effort to identify predictive biomarkers, such as the expression level of PD-L1 on tumor cells or the tumor mutational burden, to better select patients who are likely to respond. The field has moved from the simple act of releasing the brakes to a far more sophisticated endeavor to understand the entire engine of the anti-tumor immune response and to learn how to tune it with precision. Of course, such a revolutionary approach was bound to present its own unique set of challenges. The very mechanism of action that makes checkpoint inhibitors so powerful is also the source of their primary limitation. By dismantling the natural safeguards that prevent autoimmunity, these therapies risk unleashing the immune system against the body's own healthy tissues. The resulting spectrum of immune-related adverse events can affect nearly any organ system, causing conditions like colitis, hepatitis, pneumonitis, and endocrinopathies. This has necessitated the birth of a new subspecialty of clinical practice, requiring oncologists to become proficient in managing complex autoimmune syndromes, often in collaboration with rheumatologists, endocrinologists, and other specialists. This clinical reality is the inescapable consequence of Allison and Honjo's discoveries. It is the price of unleashing a powerful, previously constrained force. The other major intellectual revision spurred by this work is the complete re-evaluation of why immunotherapy sometimes fails. The focus has shifted from the absence of an immune response to the presence of specific resistance mechanisms. Research now delves into the intricate cellular and molecular composition of the tumor microenvironment, investigating the roles of suppressive cells like regulatory T cells and myeloid-derived suppressor cells, and the physical barriers that prevent T-cell infiltration. The questions have become more nuanced, moving from can we activate the immune system to what is preventing an already activated immune system from succeeding. The work of Allison and Honjo is built upon a bedrock of foundational immunology laid by giants in the field, the discoveries of the T-cell receptor by Philip Marek and John Kepler, and the elucidation of CD28-mediated co-stimulation by researchers including Carl June, provided the essential context of T-cell activation. Without understanding the accelerator, one could never have conceptualized the break. In turn, the influence of Allison and Honjo's work has been profound and transformative, extending far beyond their own laboratories. Their discoveries revitalized the entire discipline of cancer immunology, inspiring a new generation of scientists and clinicians. Researchers like Drew Partle, who was instrumental in the early work on ipilimumab, and the aforementioned Gordon Freeman and Arlene Sharp, whose elegant studies helped fully delineate the function of the PD-1 pathway, made indispensable contributions that built directly upon the central concepts established by the two pioneers. The success of checkpoint inhibitors has single-handedly launched multi-billion-dollar pharmaceutical franchises and fundamentally altered the standard of care for more than a dozen types of cancer. It has forced a complete rethinking of clinical trial design and has become the backbone upon which countless new therapeutic combinations are being built. In conclusion, the scientific journey of James Allison and Tasuku Honjo represents far more than the development of a new class of drugs. It represents a fundamental shift in the conceptual framework of oncology. They rerouted the focus of therapeutic intervention from the cancer cell to the host immune system, from a strategy of external attack to one of internal empowerment. They revealed that one of the most sophisticated weapons against cancer was already present within the patient, waiting to be unleashed. Their work stands as a powerful testament to the value of curiosity-driven basic science, a reminder that the path to revolutionary therapies often begins with asking a simple, contrarian question about how the world works. By daring to investigate the brakes instead of the accelerator, they not only illuminated a core principle of immunology, but also handed humanity the new and powerful way to fight one of its oldest and most formidable diseases, transforming the prognosis for countless patients and forever changing the landscape of medical science.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.